feeding a growing global population. It's one of humanity's biggest challenges, right? And it's this constant microscopic battle against tiny invaders threatening our crops. But what if we could give plants a secret weapon? Something really clever, something that uses the enemy's own tools against them. Today, we're taking a deep dive into a groundbreaking new strategy in plant immunity. We're unpacking some really fascinating research from Junzu Wang and colleagues just published in Nature. Our mission really is to explore how scientists are fundamentally remodeling plant defenses, trying to create broad spectrum and remarkably durable disease resistance.
Yeah, this research, it offers a genuinely innovative approach. It could really significantly reshape how we protect our food supply.
Yeah.
Especially from the ever-evolving threats of plant pathogens. It feels like, potentially a real game changer.
Okay, so let's unpack this. When we talk about plant defenses, what are we actually talking about? How do plants naturally guard themselves against diseases and pests? What do their sort of built-in security systems look like?
Well, at their core, plants have an innate immune system. And central to this system are these special proteins called NLRs. That stands for nucleotide binding and leucine-rich repeat immune receptors. Bit of a mouthful.
Right, NLR.
Yeah. Think of them as like internal security guards. They're constantly patrolling for specific molecules, effector proteins that invading pathogens release. The moment they detect one, bam, they trigger powerful defense responses.
Okay, like what kind of responses?
Often it includes what's known as the hypersensitive response, or HR. That's where cells right at the infection site basically commit programmed self-destruction.
To kind of quarantine the threat, stop it spreading.
Exactly, to wall it off.
That sounds incredibly effective, really sophisticated. So if plants have these amazing systems, what's the catch? Why do we still face such, huge crop losses to diseases around the world?
The challenge, well, it lies in their specificity. A
Yeah.
single NLR typically recognizes only a very, very limited number of these pathogen effector proteins.
Oh.
It's like having a highly specialized lock that maybe only fits one or two very specific keys.
Right. So not very flexible.
Not at all. It means the plant's natural resistance is often what we call narrow spectrum, or race-specific.
Hmm.
And that's easily bypassed as pathogens, which evolve incredibly fast, develop new effector proteins, new keys.
So it's a constant arms race.
Pretty much, yeah.
Mhm.
An ongoing evolutionary battle.
That makes perfect sense. So given this battle, scientists have obviously been trying to, upgrade plant immunity for a long time. What have some of the earlier strategies looked like, and where did they sort of run into limitations?
Yeah, there have been various attempts, things like site-directed mutagenesis. But one particularly clever strategy is called decoy engineering.
Decoy engineering.
Yeah,
right?
imagine the plant has a specific tripwire, but it's not meant to be triggered normally. Scientists basically modify a normal plant protein, let's say one called PBS1, and turn it into a decoy tripwire. The really ingenious part is a pathogen's own enzyme, a proteases it secretes, is what actually cuts this decoy. And when that cut happens, that action springs the real trap. It activates a specific NLR protein like RPS5, and boom, triggers immunity.
That is a fascinating concept, really turning the pathogen's weapon against itself. So what were the main roadblocks then? What kept these earlier methods from being, widely adopted or truly broad spectrum?
Well, decoy engineering, while it's clever, it's technically quite demanding. You have to successfully engineer both the decoy protein and find or engineer a specific companion LR, like that RPS5.
Ah, right, two components.
Exactly. And the problem is many important crop species, they simply lack RPS5 or similar helper NLRs. That severely limits where you can even try it.
I see.
Plus, just like natural resistance, these engineered decoys, they can still be outsmarted by rapidly evolving pathogens. They often lack that true broad-spectrum activity or, long-term durability out in the field.
Okay, so that brings us to this paper. This is where it gets really interesting, you said. It presents an entirely new, and you called it elegant, solution. What's the core idea? What's the innovation here with remodeling these NLR?
The key is to engineer what they call an autoactive NLR, or ANLR. These are NLRs that, if left alone, would be always on, constantly triggering defenses.
Which wouldn't be good for the plant, right? Too much energy drain.
Exactly. Constant defense activation is costly. So the clever part is they attach a flexible blocking peptide right onto the beginning of the protein chain, the N-terminus. And crucially, this blocking peptide contains a specific pathogen-originated protease cleavage site, or PCS.
So it's like a safety cap. The NLR is inherently on, but this peptide blocks it.
Precisely. That N-terminus is essential for many NLRs to function. Attaching this peptide Basically handcuffs the NLR, keeps it inactive.
Okay.
But here's the trap. When a specific pathogen invades, it releases its proteases, its molecular scissors. These proteases act like a specific key to the lock of that PCS on the blocking peptide.
Ah, so the pathogen's own enzyme unlocks the defense.
Exactly. It cleaves off the blocking peptide, releasing the now free ANLR, which instantly switches to its on state and triggers a strong, effective, innate immune response.
Leading to broad-spectrum resistance.
And hopefully durable resistance, too. Yes.
That mechanism, it sounds remarkably elegant. It really does. So just stepping back for a second, how do they actually make an NLR autoactive in the first place? How do they flip that switch?
It's actually relatively straightforward now for scientists. They can construct an ANLR by making a small but critical change. It involves introducing specific amino acid substitutions in a particular part of the protein called the MHD motif.
MHD motif. Okay.
This motif is known to be crucial for regulating the NLR's on-off state. So changing just one amino acid there, maybe swapping an aspartate for a valine, can effectively jam it in the on position.
Until you add that blocking peptide.
Until you add the blocking peptide with the pathogen-specific cleavage site.
Exactly. That's a fantastic explanation. So how did they put this concept, this ingenious idea, to the test in the lab?
Right, so they focused on potyviruses.
Potyviruses, you mentioned those.
Yeah, it's a huge family of plant RNA viruses, responsible for something like 30% of all known plant viruses. Includes major agricultural threats, like potato virus Y, PVY, big problem for potato growers.
Got it.
A key insight here is that potyviruses encode proteases, enzymes like NIA, and importantly, the sites these proteases cut are highly conserved, very similar across many different potyviruses.
Ah, so one target site could work against many related viruses.
That's the idea. It makes them an ideal target for trying to achieve broad-spectrum resistance. So they engineered a specific protein. Let's call it the PVY immune trigger for simplicity. It combined that blocking peptide, a conserved PVY cleavage site, PCS-PVY, and an autoactive CNL, which is a type of NLR called TM22.
Right.
Initially, this engineered protein, as expected, was inactive. But when they co-expressed it in plant cells with the PVY protease...
The protease cut the blocker.
Exactly. It was cleaved, releasing the autoactive TM22 and triggering cell death, the HR response, precisely as they designed.
Okay, so it worked in cells, but did this PVY immune trigger actually protect whole plants from the virus itself?
It did. And quite dramatically, too. They did experiments with Nicotiana benthamiana plants, a common lab model. This engineered TM22 conferred what they described as classic complete resistance against PVY.
Complete resistance. Wow.
Yeah. You could see the visible HR lesions, those localized cell deaths, on the leaves they inoculated. But crucially, the rest of the plant, the systemic leaves, were completely free of viral symptoms, and they couldn't detect any viral RNA.
That's impressive.
And even more impressively, because that cleavage site is so conserved across the potyvirus family.
Right, you said it was conserved.
This single-engineered NLR also provided broad-spectrum and complete resistance against several other potyviruses, things like turnip mosaic virus, TUMV, plum pox virus, PPV, pepper mottle virus, PPOV, and chili venal mottle virus, TRIVMV.
So one engineered gene protects against a whole suite of related viruses.
That's what they found. And they didn't stop there. They also showed this works with RNLs, which are another major class of NLRs. They use one called ATNRG 1.1, engineered it the same way with the PVY cleavage site. This also showed broad-spectrum resistance. And interestingly, in this case, it often conferred what they called extreme resistance.
Extreme resistance? How's that different from complete resistance?
It means the plants develop totally normally. No visible HR lesions at all, and still no detectable viral RNA anywhere. Like the virus couldn't even get a foothold to trigger that visible HR.
That's incredible. The scale of that protection is really significant. One protein against multiple viruses in the same family. But, what if a plant needs to defend against viruses from different families, ones with different protease cleavage sites? Can this strategy be expanded? Can you make it even broader?
That's a great question, and it's exactly what their researchers looked into next. They noticed that their initial PVY immune trigger was actually susceptible to another virus, tobacco itch virus, or TEV.
Ah, okay. What?
Because TEV's protease cleavage site is different from the PVY one they used. The key didn't fit the lock.
Makes sense.
So this led them to investigate using tandem cleavage sites, basically putting two different PCSs, two different locks, onto the same blocking peptide.
Like adding another notch to the keyhole.
Kind of. They created a new engineered protein, this time incorporating both the PVY and the TEV cleavage sites, one after the other.
And did that work?
It did. This new protein, with the tandem sites, was recognized and cleaved by both the PVY protease and the TEV protease, and both triggered immunity.
Fantastic.
So it demonstrates that using these tandem PCSs can indeed expand an NLR's recognition specificity. You can confer a broader spectrum of disease resistance. Now, they noted that for TG, some plants showed only partial resistance with this setup, but it definitely expanded the protection significantly beyond just the PVY family.
So it's effectively like creating a kind of master key system where multiple different pathogen proteases can unlock the plant's defenses.
That's a great analogy, exactly. And what's really fascinating here, technically speaking, is that the engineered ANLR seems quite robust. It can apparently tolerate having a short peptide fragment, up to maybe 8 amino acids, left hanging off its N-terminus after cleavage without severely impacting its ability to trigger immunity.
That suggests some flexibility, maybe for adding even more sites later.
Potentially, yes. It opens up possibilities.
Okay, this all sounds incredibly promising in the lab setting with model plants. But, for global food security, the real test is getting it to work in our actual crops out in the field. Did they manage to take this step, move from Nicotiana benthamiana to a real agricultural crop?
They did. That's a crucial step, obviously. To test its applicability in a major crop, they generated transgenic soybean plants. These plants expressed a similar engineered protein based on that ATNRG 1.1 RNL, but this time they incorporated a cleavage site specific to soybean mosaic virus, SMV.
SMV, a major threat to soybeans, right?
Absolutely. A very common and damaging virus for soybean production.
And what were the results in soybean? Was it as effective?
The results were hugely successful, according to the paper. The transgenic soybean plants exhibited complete resistance to SMV.
Complete resistance in soybean? That's huge.
It is. And crucially, these resistant plants, they grew and developed normally. Their traits like height, biomass, seed setting, they were similar to the uninfected wild-type soybeans.
So no obvious yield drag or fitness cost, at least in the greenhouse.
Exactly. That suggests no significant growth trade-offs under those greenhouse conditions. And that's vital if you want to translate this strategy into practical agriculture.
Absolutely. That's a massive leap forward towards actual crop protection. So what's the next critical step after successful greenhouse trials like these?
Well, while the results are highly promising, the researchers themselves noted the sample size in their initial soybean experiments was relatively small. So the next essential step really have to be extensive field trials.
Right. Real-world conditions.
Exactly. Those are crucial to rigorously evaluate the agronomic performance, make sure yields hold up, check for any unexpected fitness cost in real agricultural environments, see how it performs outside the controlled lab setting.
Okay. So pulling all this together then, if we look at the big picture, what are the biggest, most impactful takeaways from this deep dive? What should we remember about this for the future of plant health and ultimately global food security?
I think the central insight is just how simple and remarkably effective this strategy appears to be for engineering disease resistance. Compared to previous methods like decoy engineering, it's simpler because you potentially only need to engineer a single gene, a single NLR.
Right, just one component instead of two.
And its applicability seems incredibly broad. It could potentially be used in any crop plant that naturally encodes one of the hundreds of known CNLs or RNLs, which is most plants.
So potentially universal applicability.
That's the hope. And the resistance it confers seems genuinely broad spectrum. As we discussed, a single engineered NLR could protect against, potentially over 110 different podiviruses just by targeting that one conserved cleavage site. And with tandem cleavage sites, the spectrum could broaden even further.
And what about durability? That's always the big question with resistance, right? Pathogens are constantly evolving. Will they just find a way around this too, eventually?
This is where the strategy gets particularly exciting and potentially quite unique. The engineered resistance is expected to be highly durable.
Why is that?
Well, think about it. For a pathogen to overcome this resistance, it would need to develop mutations in its own essential proteases, the very enzymes it relies on for its own replication and survival.
So it'd have to change the key.
But changing the key might break its own engine. Mutations that stop the protease from cleaving the engineered site would very likely also disrupt the protease's ability to process the pathogen's own vital proteins,
making the mutation potentially lethal or at least severely damaging to the pathogen itself.
Exactly. It's a kind of biological catch-22 for the pathogen. It creates a very difficult evolutionary hurdle for them to overcome, much harder than just tweaking an effective